Hello, welcome to 13 Universal. We are still solving A-level CIE biology, and this is paper 5, planning, analysis, and evaluation. Paper 5, okay? Get your writing materials. Let's dive into these waters. Let's go for a big catch, right? The first question, remember we solved together, right? So yes, see the first question there. When bananas, I like bananas. When bananas ripen, they turn brown in color. This is due to the formation of a brown product called melanin. Wow, banana has melanin too, like human. Amazing. The enzyme catechol oxidase acts on its substrate. So catechol oxidase is an enzyme. It acts on a substrate called catechol. So catechol is a substrate of catechol oxidase. Note that. Leading to the formation of a brown product, melanin, as shown in Fig 1.1. That's Fig 1.1. This is catechol, pale pink substrate, acted on by catechol oxidase to form melanin, brown products. Okay, amazing. Now there we go. Make sure you remember this because that's a massive reading. As melanin is produced, the color of the reaction mixture changes to brown. The intensity of the brown color produced is proportional. Watch this. As melanin is produced, the color of the reaction mixture changes to brown. It turns brown. The intensity of the brown color produced is proportional to the concentration of melanin. Okay, that means that as melanin concentration increases, the intensity increases, right? That means melanin concentration increases because the rate of reaction is high. When the rate of reaction is high, melanin concentration will increase, then intensity of the color will increase. The more active the enzyme, the more intense the color. See it? So the more active, a colorimeter is used to measure the absorbance of the reaction mixture. Absorbance is a measure of the light absorbed by a color solution. With the reaction shown in Fig 1.1, the more intense the brown color, the higher the absorbance. See, the more intense, the higher the absorbance. That is, the more light is absorbed, more light wavelength absorbed. Catechol oxidase can be extracted from banana, so we can get it from banana. Let's go on. Some students were asked to extract catechol oxidase by grinding a slice of banana with sand and water using a mortar and pestle. The resulting mixture was then filtered through a piece of cloud. Suggests a reason: number one, for grinding with sand; number two, filtering through cloud. All right, pause the video and try to answer the question: why do we have to grind bananas with sand? Okay, I think the reason is to break up the cell in order to release the enzyme. The enzyme is inside the banana cells. Okay, so we need to break up the cells, right? Break up the cell to release the enzyme, and we filter to remove the sand. You need to remove the sand, okay, or cell debris, right, so that we can have pure enzyme, right? See that? Grinding with sand to break up the cell, release the enzyme, filtering to remove sand debris. In the wording, 2 marks. The students investigated. So watch this. The students investigated the effect of the substrate concentration on the enzyme catalyzed reaction shown in Fig 1.1. So they investigated the effect of substrate concentration on. So which of them is the independent variable? Which one is affecting which one? Okay, these are the things you have to take note of. So which one? So substrate concentration is affecting enzyme catalyzed reaction. All right. So substrate concentration is independent. Why this is dependent? This is going to be measured. This is going to be changed. All right. Let's see how it worked. The students were provided with the extract catechol oxidase enzyme solution prepared in step A, which was kept cold. So see, it has to be kept cold. Every information you are given is a clue. You're going to be needed to use it again to plan an experiment. So it has to be kept cold until needed, so that why does it have to be kept cold? Because it's temperature sensitive. All right. So a stock solution of 1% catechol solution. So catechol is a substrate made up in a buffered solution of pH 7.0, a buffer solution of pH 7.0. So why do we need a buffer? To maintain pH, okay? Because enzyme is pH sensitive. So this should be the optimum pH, all right? 7.0. All right, let's go. The procedure used by the students is outlined in step 1 to 3. Number 1, the extracted catechol oxidase enzyme solution was mixed with 1.0% catechol solution. Okay, so they mix the enzyme and its substrate. All right, 1.0%. Okay, after 2 minutes, a calorimeter was used to measure the absorbance of this mixture. 3. Step 1 and step 2 were repeated using different concentrations of catechol solution. Okay, the first question here suggests why the students use a colorimeter to measure the absorbance rather than judging the intensity of the color by eye. So why do we use colorimeter, okay, instead of using our eyes? Okay, so pause the video and write it down. Okay, suggest. I think the reason is because colorimeter gives us a numerical value. Okay, it's objective, it gives us, all right, unlike the eyes is subjective. Okay, so this gives us a numerical value. All right, see it here. So result is quantitative, that's numerical. All right, AW, any other wording. Okay, so we use it because it gives quantitative, numerical, subjective. Okay, value, numerical, quantitative value. The eyes gives subjective value. Okay, all right, that's 1 mark. Yeah, identify the independent variable and the dependent variable in this investigation. So what is the independent variable, guys? What are we changing? What are we changing? Okay, independent variable is what they're changing, which is the concentration of catechol. Okay, that's the independent variable, concentration. But dependent variable is how you are measuring, what you are measuring is the enzyme catalyzed reaction. But how are you measuring it? All right, absorbance. You're measuring the absorbance. All right, so absorbance is then the dependent variable. Okay, and this concentration of catechol, which is the substrate. All right, let's go. 2 marks. The students decided to investigate the effect of inhibitors. So it's now changing the dynamics of this question. The effect of inhibitors on the reaction shown in Fig 1.1. The students plan to add an inhibitor Y to reaction mixtures containing different concentrations of catechol solution. Describe a method. A method is always coming out. 5 marks, 6 marks, 8 marks, 7 marks. That's a lot. Sometimes 9 marks. All right. You have to master it. The method the students could use to collect the data needed to test the effect of inhibitor Y in reaction mixtures, okay, containing different concentrations of catechol solution. The description of your method should be set out in a logical way and be detailed enough for another person to follow. You should not repeat details from, all right, A, describing how the extract of catechol oxidase is prepared. So we don't need to work on how catechol oxidase is prepared, grinding up banana with sand and water and then filtering. Okay, it's not necessary. Okay, so pause this video and try to, describe it. Write an essay, produce a descriptive essay of this experiment, these procedures. Okay. All right, like I said, you will always have to identify your independent variable, and independent variable is how you're going to vary it. Okay, so before you start this essay, how you gonna, all right, you will need to write what is it called, an outline. So this is an outline here. So in my outline, I'm figuring out, okay, before I start this essay of 8 marks, 7 marks, I need to plan it. Okay, what am I going to do first? I say, okay, my independent variable, I'm changing. All right, vary means change. How am I going to change it? How am I going to vary it? Then I'm going to go first and talk about, of course, dependent variable. Right, dependent variable is what I'm going to measure, right, which is of course dependent variable here is absorbance, independent is concentration of catechol. So you're going to measure, this is measure, all right? So yeah, that's dv is dependent variable, and kv, so iv dv kv. Okay, so you have to, that's part of your planning. So this is kv is key variables, all right? You need to keep constant. What are the variables that can actually affect the rate of reaction, all right, that I need to keep constant? So you have to note them before you start writing. This is your plan before you jump into it. And then you have to know how to ensure reliability. Reliability, you have to replicate and calculate the mean. So reliability is very important. And then apart from reliability, you have to think about hazards, right? What are the hazards? Yeah, and risk, precaution. Yeah, so now that is my outline before I start writing. Okay, so I'm going to talk about my independent variable here is the substrate concentration, which is catechol. Okay, so I need to vary it. How do I vary it? Dilution. Okay, so there's a description of your method should. So dilution. So I have of course, from what they said there, look at what they said. Investigate the effect of the students plan to add mixtures containing different concentration of catechol. So you need to produce the different concentrations of catechol. Remember that the stock solution is 1. So I need to state 5 values, 5 concentrations between 1 and 0. Okay, 4 between them. 1 is inclusive. So now I have 1. Okay, the interval, I can decide to choose 0.8, right? 0.8, the interval is 0.2. So the next one, 0.6, 0.6, and then 0.4, 0.4. Okay, and then we have 0.2, 0.2. Yeah, okay. All right, so now these are produced using dilutions. Okay, so dilutions are made. All right, of course, simple dilutions made using stock solution to produce this concentration. So solution, so I start writing that the solution, all right, of catechol, the same volume of catechol actually is of different concentration of catechols added to test tubes, all right? But meanwhile, you should have noted your dependent variable, which is absorbance, and key variables here, volumes of all the solutions should be the same, okay? And concentration of the enzyme should be the same, right? Temperature should be kept constant, and of course pH using the buffer, temperature using um, the water bath. Okay, so all of this, so you go on and write. Okay, you just describe it. All right, you take, all right, same concentration like they said, same concentration. First of all, you extract the enzyme catechol oxidase, put it in cold condition, and then of course take same volume of the enzyme, put it in different test tubes, and then take same volume of course put same volume of the substrate catechol into test tubes, and then equilibrate the temperature of both the enzyme and the substrate. Put them in different test tubes and then expose them to the same temperature, equilibrate them, and after equilibrating them, you can mix the enzyme catechol oxidase and the substrate catechol, and allow the reaction to occur for 2 minutes. All right, and then you now measure the absorbance by putting it into a colorimeter. Make sure you reset the colorimeter as the colorimeter is reset. All right, reset the wavelength using a filter, a color filter, and then reset the absorbance using water, distilled water. Okay, so measure the absorbance for each concentration, carry out different, of course. Meanwhile, you're going to have to add the inhibitor in each, the same concentration of the inhibitor. Is that okay? All right, of the inhibitor. All right, I want to check the effect because they want to determine the effect of inhibitor, right? So of course, by the time you're done with that, you want to check what is the effect of the inhibitor, and then you also measure the absorbance, then calculate the mean for each one, determine about 3 replicate values. Then that's for reliability. Your hazard, the enzyme could be an allergen or irritant, so you wear gloves. Okay, so look at it, guys. Using at least 5 concentration of catechol, so use, all right, and then state range 1 between 1 and 0, I stated it there. Okay, then enzyme solution, keep it cold until when it is needed. So the enzyme and substrate, you have to equilibrate 2 of them before mixing them, so you have to write all of this. So keep the reactant or reaction mixture at constant controlled standard temperature, so because temperature has effect on enzyme catalyzed reaction. All right, so same known volume of catechol, substrate, and all of that. See, number 7, same constant standardized known volume of catechol, same known volume of catechol oxidase. So same volume of both substrate catechol and enzyme catechol oxidase. That's very important. 9. So measure the absorbance intensity of color. Okay, one set time. Additional details of calorimeter, how to reset calorimeter, set the wavelength and the absorbance. All right. So this is repeated. See, number 11, repeated how many times? Twice or 3 times. Calculate the mean. That's for reliability. Idea of repeating whole experiment. Now you have to repeat the whole experiment without the inhibitor. So the first one with the inhibitor, the whole experiment without the inhibitor. You now compare them, okay, to check the effect of the inhibitor on the absorbance, which represents rate of reaction. So name hazard and then precaution. Example, enzyme could be an irritant, all right, so you wear gloves, so it's a hazard. Okay, the temperature also wear hand gloves. Okay, so that's 8 marks. You could produce card. Now look at this. Fig 1.3 shows results when no inhibitor Y was added. Okay, all right. The students suggested that inhibitor Y was acting as a non-competing inhibitor. Okay, it says on the sketch, show sketch on fig 1.3, this fig, sketch the curve expected if inhibitor Y was acting as a non-competing inhibitor. All right, the knowledge of this topic is very important. So non-competing inhibitors, all right, always lower the rate of reaction from very beginning. So it's going to be, and it's going to lower the rate of reaction. Okay, so it levels off, but the Vmax is different. Okay, so this is how non-competing inhibitor, the graph shows. Okay, so you see percentage concentration, as you increase subject concentration, it also increases but does not reach the Vmax, it levels off. Okay, so that's how to sketch it. So say curve should be on the right side of this original curve, this right side of the original curve. Should plateaus below the original curve, so it also plateaus. So for being on the right side, 1 mark. For plateauing, and that 1 mark. Somebody asked a question. I heard you say, okay, what about if they said, what about if it is competing inhibitor? How would it appear? If it's competing inhibitor, remember that if it's competing inhibitor, right, it is going to, or should I use this? Okay, this is the graph without inhibitor. If it's competing inhibitor, as you increase of the concentrations, it will increase and then reaches, okay, the same Vmax and then levels off. So in other words, competing inhibitor will appear differently when you sketch the graph compared to, all right, non-competitive inhibitor. Okay, so that means if you're going to do for competitive, it's going to increase and reach Vmax and levels of. That's for competitive, but this for non-competitive inhibitor. Okay, so 2 marks for that. All right, look at the next question. They said Vmax is the maximum initial rate of reaction of the enzyme. The Michaelis-Menten constant, Km, is the substance, sorry, this is substrate. It should be substrate concentration at which the initial rate of reaction is half, all right, its maximum value. Draw on fig 1.3, the position of Vmax and Km of the enzymes when no inhibitor Y is present. Use your graph to describe the effect of the addition of Y on the Km. Okay, so to get the Km, remember that Km is the substrate concentration, like we said. All right, that's substrate concentration at half Vmax. Half Vmax. So, so first of all, you have to calculate your Vmax, and then determine the half of the Vmax. So that's substrate concentration at half Vmax is your Km. Okay. So Km is the maximum. So this is our Km. Let's trace it. Trace it. Okay. Trace it. Sorry. Trace this line. Yeah. Come with me. Trace it. Trace it. Trace it. Yeah. Trace this line. So this is the Vmax. That's the maximum. So this is the Vmax. Okay. This is the Vmax. How many boxes? From the beginning. Okay. How many boxes? Right. From here to here is 10. Okay. That's like 18 boxes, right? Yeah, that's 11, 12, 13, 14, 15, 16, 17, 18. So half Vmax should be used to determine Km. So half Vmax is 9 here, right? So I'll come here, right? This is the ninth line, ninth box. Okay. So this is Vmax. This is Vmax, that's the maximum. Okay. That's Vmax. Vmax. Right, this is half Vmax. Half Vmax. Half Vmax. Okay. Yeah. So just trace it. Trace it to the curve. When you get to the curve, so this is it. You now trace it down, extrapolate it. Okay. So the substrate concentration at this half Vmax is our Km. So we are going to see our Km. So that's our Km. So remember, this axis is for percentage concentration of category, which is substrate. So this is Km. Is that okay? Yeah. So Km is substrate concentration at half Vmax. All right. So let's see. See, correct placement. Yeah, we've done well. So correct placement of Vmax and Km. 2 marks for that. Oh, we didn't mention. Okay. So you see, it's going to have the second question. For non-competing inhibitor, you to have the same Km value. Yes, with the graph, all right, that has no inhibitor, right? So the same Km value. See, the non-competing inhibitor decreases the Vmax, all right, but same Km, right? So, but if it is competitive, same Vmax, but increases the Km, right? Yeah. So I think I should help you with that, right? Let me help you with that. All right. Let me help you with that. Watch this. Okay. Watch this table. Sorry. Yeah. Let me help you with that. So this is, let's assume this is Vmax. Okay. Vmax. Vmax. Let's make it big. Vmax. Yeah. That's a big guy. Right. And this is Km. Km. Okay. Yeah. Okay. Now this is competitive inhibitor, CI, competitive inhibitor. Okay. That's the first rule. Okay. So the effect. Now this is non-competitive inhibitor, second rule. N is non-competitive inhibitor. Okay. So let's see the effect it has. So Vmax for competitive inhibitor, same without inhibitor, same. Okay. Don't forget this. Now Km, right? Km is higher. Okay, yeah, the Km is higher or increases if you use complete inhibitor. That means that the enzyme activity will be low because Km is indirectly proportional to enzyme activity. Now for this, okay, this lowers, okay, it decreases, that's as lower Vmax, right? If you use non-competitive inhibitor, the Vmax will be lower, all right? It decreases it. Why the Km will be same, okay, when compared to the reaction without inhibitor, okay? So don't forget that. So this is why when compared to yeah, has same Km value than with no inhibitor. That is this. Km is same. Okay. All right. So hypertension is a condition where people have long-term high blood pressure. These are long-term blood pressure. Two main types of drug are used to lower blood pressure in people with hypertension. This is ACA inhibitors, diuretics. Scientists investigated the effectiveness of drugs when separated and in combination. 505 people with hypertension were split into 4 groups as shown in table 1.1. 4 groups. How many of them in the group were not told? But this is group 1, placebo, 2, ACA inhibitor, 3, diuretic only, 4, ACA inhibitor and diuretics. Okay, so results are shown in fig 1.4. Alright, so this is the result. So if you are asked to discuss this result, what are you going to say? What are you going to say? Just pause the video and discuss the results. If you're asked to discuss it. Okay? Of course, you can tell them based on this result, right, that group 4, which is ACA and diuretics, ACA inhibitor and diuretics, gives the highest or the largest, okay, mean reduction in blood pressure. Okay? That makes sense. But we cannot count on this. Okay? However, there are no error bar, so we don't have error bar to determine whether the mean reduction in blood pressure actually overlap. So there's no statistics test to use, so we can't count on that. Okay, so because when they say you should discuss, you should talk about, all right, give information, okay, on the both sides of the argument. Okay, so if we're going to use that, this is the best to use, of course, because it gives the largest. But why can't we use this, just the opposite side of it? Because there's no error bar to determine whether this and this are not significant difference, and we don't have any statistic test, all right, to prove that this might be the best treatment. And again, guys, you know that when you're looking at this, you find out also they did not give us information, the initial blood pressure and the final, so we don't know the percentage decrease from the original. And of course, if you look at it, we don't actually need this, of course, to reduce blood pressure. We can use group 1, which is placebo. All right, so, and we don't know the side effects or long-term effects of using group 4 ACA inhibitor and diuretics. Okay, so the scientists analyzed the data and concluded that a combination of an ACA inhibitor and the diuretic should be used to lower blood pressure in the treatment of hypertension. With reference to the data in table 1.1 and fig 1.4, discuss conclusion that a combination of an ACA inhibitor and a diuretic should be used to lower blood pressure in the treatment of hypertension. Okay, you can pause the video or try it, go back to the diagram and try it. But we have already answered this question, all right? Okay, we have answered it, that the pressure, of course, you can from the diagram, from that figure, we can use ACA and ACA inhibitors and antidiuretic because they produce largest mean reduction. Okay, but there are other aspects of it which also contradicts the conclusion. All right, the fact that there are no error bars to check whether there's a significant difference or not, there's no statistical test, all right, used, and of course you check that there's no long-term effect stated. Now the placebo can as well be used. Look at it. Anytime they're asked to discuss, evaluate, and all of that. So combination of drugs, yeah. So you state what you see, and then you make uh, argument. Okay. So no error bars, no statistical test, no t-test compared, no data on long term, and no information on this. No information on blood pressure prior to trial. No information on type of blood pressure. No information on number of patients in each group, no data site. Now all of this from here down is trying to discuss the conclusion that the combination. So it's trying to prove that, of course, this particular conclusion that AC inhibitors and diuretics should be used to lower blood pressure. So this is the only one that supports it. So as a scientist, upcoming scientists, you have to be objective. All right, look at the two sides of the argument, and then that's when they say discuss, that's what they look at. Okay, all right, be objective. So that is it, 3 marks, any three. Okay, this is number 2 question. Seems is inheritance. Yeah, do you like inheritance? Inheritance of wing shape and eye color. These are 2 genes, wing shape and eye color. That's dihybrid. In fruit fly, Drosophila melanogaster is controlled by 2 genes. I said it. Gene, yeah, these are 2 genes, the 2 alleles. This is capital, controls wing shape. Allele N, uppercase, for wrinkled wings is dominant to allele lowercase for normal wings. You can see that's recessive. See the second gene, gene uppercase E, all right, and that of these are 2 alleles, controls eye color. While the uppercase for rosy eyes is dominant to allele E for red eyes. A biologist predicted that that's what should be, that if genes are on different chromosomes, if they're on different chromosomes, the ratio of the phenotype of the F2 generation would be 9331. That's what it should be. Okay, the guy predicted that. The biologist went out to research, okay, whether that would work for this. The biologist carried out a breeding experiment. Homozygous dominant fruit flies with wrinkled, okay, these are pure breeding, true breeding. So homozygous dominant fruit flies with wrinkled wings and rosy eyes, the dominant were crossed with homozygous recessive fruit flies with normal wings and red eyes. All the F1 fruit flies had wrinkled wings and rosy eyes. Amazing. The F1 fruit flies were crossed with each other. All right, that's to produce F2. Table 2.1 shows the result of the F2 generation. Okay, remember that what we're working on here based on the knowledge of the syllables of A2 and AS. Okay, so to be able to understand what we're doing, you should be acquainted with that syllabus. Look at the F2 generation. Okay, can you see, what do you observe here? Okay, wrinkled wings and rosy eyes. This is dominant. This is a parental class, the first parental class. All right, 44. Okay, now look at another parental class. All right, so the ones in the middle, which are the recombinant classes, okay, due to crossing over, due to crossing over, there are 2 this doesn't follow this. Okay, so from the result you might see, because that result said that the ratio should be 9, okay, 3 3 1. So if you try to do the ratio, it will not follow this. So one starts thinking whether these 2 chromosomes, whether they are actually on different chromosomes. If they're on different chromosomes, they will give this ratio. Are they really there? Okay, we're going to find out. That's what I want to find out. We're going to find out, right? So if you divide everything here by the smallest, which is 2, right, the ratio, if you say 44 divided by 2, that will give you 22. That's quite 9. This will give you 1 1, and this will give you 8, and it doesn't follow this. Okay, so let's find out what happened. Okay, let's go. And of course that's 64, the total. Now yeah, the chi-square test was used to analyze data in table 2.1. State one reason why chi-square test was used. Why do we use chi-square? Pause the video, write the answer down, pause and try. Okay, why do we use chi-square? Yeah, we use it to check to determine whether there is significant difference between expected result and observed result. Okay, and we use it when the data is discrete. Okay, discrete, all right, discontinuous. Discrete means that each of the measurement, all right, so belong to a particular group. Okay, so is the data is discontinuous, that means that it's not, the data is not within, it's not between two extremes. Okay, so that's when we use this. Okay, and then it's of course nominal. So look at it here. Idea of compare expected and real ratio. So anytime you are dealing with theta chi-square, remember the data should be categoric, right? It should be categoric. Different types, they're different categories. So like the phenotype gives a different categories. So the data for each of them represent a particular category. So it must be discrete, discontinuous, nominal, and binary, just no sequence, 1 mark. State the null hypothesis that the biologists would use for this test. So state it. Null hypothesis, pause the video and try, is a negative hypothesis. Okay, is a negative hypothesis. So it just says there's no significant difference between the expected result and observed. Yeah, that's what it says. No significant difference between the observed and expected result. The difference between expected result is due to chance. Okay, that's what it's trying to say. There's no significant difference. So it's due to chance, but use the first one. Okay. All right, 1 mark. Complete table 2.2. Calculate the value of x chi-squared for the result of the F2. This is F2 generation. Okay. Now that's the equation for that. I want you to pause the video and calculate this. Okay. Pause and try this, because you have to get the total here. Yeah, what's my total for that? If I add up this 44 plus 2 plus that's 48 plus 16, that's 64. Okay, so if I say 48 plus 16, that's 64. Yeah, so total here is 64. So I needed to calculate the expected result. Expected result is gotten from the ratio, right? So this is, remember that the ratio of this should be 9:3. Okay, is to 3:1. So total ratio is 16. So if I want to get this one, I will now say 9/16. Okay, 9/16. Use the ratio to calculate the expected. You must always use the ratio to calculate the expected, times total 64. Okay, so 16 divided 64 will give you 4, right? So 64 divided by 16 will give you 4. 4 times 9 is what? 36. So the expected number for the wrinkled wings rosy eye is 36, based on this, right? That's expected. So I want to do this the same thing. I'm going to use the ratio 3/16. 3/16 times 36. So work it out. This is times, multiply 36. That is 4 times. Now because 16 goes into 36, 4 times that's going to be 12, right? 12. Yeah. So the same thing here. 13/16. 13/16. Yeah. That's 16, please. Times 36. Okay. Yeah. So that will give us the same 12. All right. 12. And this will give us 4. Okay. Use this, and that's 1/36. 1/16. Sorry. 1/16 times 36. No, times 64. What am I saying? This is 64. Okay. Please correct it. 64. That's the total. 64. Yeah, 64. That's 64, please. Yeah. 64 is the total. That's what we're using. 64. Okay. Yeah. 64 is total of the observed result. Okay, so that will give us 4. Yes, so yeah, 4. So we are done with the first column, guys, but we have the second column to deal with. So this is this minus this. Okay, that's observe 44 minus 36 divided by 36. So let's do it. So pick up your calculator. Okay, observe is 44, expected is 36. So 44 minus 36 gives you what? 8. 8 divided, all right, by 36. Okay, so all right, so just to help someone, just say 44. I'm just going to do it once. Minus, that's observe, minus 36 over expected, okay, which is 36. Okay, that's for once. All right, so we're going to do that. So that's 8 divided by 36, that gives us 0.2222. so the answer here should be, all right, 0. Right, 2 2 2. So do the same for the next row. Okay, this is 2 minus 12 divided by 12. So if you say 2 minus 12, oh, we forgot to square. Oh, we should, whatever we'll get to square before dividing, we forgot to square. How could I forget to square? How could I? So you have to square, see? Yeah, so everything here is squared before you divide. Okay, yeah, so let's see what we get. You have to square everything there. So let's do it again. 44 minus 36, 8 squared is 30. 8 squared is what? So that's 64. Is that okay? 8 times 8 is 64. Then 64 divided by 36. What does it give you? It gives you 64 divided by 36. That will give you 4, right? Yeah. Okay. No? It will give you 1.7. Yeah. 1.7777. So 1. Okay. 7, maybe 8. You can put 8 here. Okay. 3 can figure. Okay, let's do this. The next one quickly. So that is 2 minus 12 divided squared and then divided by 12. So 2 minus 12 will give you 10. 10 squared is 100. 100 divided by 100 divided by 12. Okay, 100 divided by 12. We are dividing everything by 12, right? Yeah. So that gives you 8.333333. Yep. So this will give you right, 8.333. All right, let's go to the next one. Remember, observe, which is this, minus expected all squared. So that's 2 minus the same thing. 2 minus 12 is minus 10. Minus 10 squared is 100. 100 divided by 12 will give you the same value here. Okay, negative square will give you positive, right? 8, 3. Okay, 3, 3. So the same thing here. 16 minus 14 will give you 12. Okay, 12 squared is 144. 144, because we are following this. Okay, this minus this will give you 12. All right, 12 squared, 144. 144 divided by 4. 144 by 4. Okay, that's 36. So we have 36 here. So let's add up everything. Okay, because chi-square calculated is at the summation of all of this. So we have to add everything in that column, everything here. So bring your calculator, let's do it if you have not done it. So that's 1.778 plus 8.333 plus 8.333 plus 36. What does it give you? It gives us 54.444. Okay, 54. So that gives us 54. Yeah, 54 point what? 0.444. This is chi-square calculated. 54.444. Yeah. So there it is. Look at the answer here. We got it. Okay. Yeah. See. So if you have question, let us know. Just follow the calculation. All right. Just one mark. All of this. That's too much. Too small. Table 2.2 shows some critical value of chi-square at different probability levels. And as a state why the student used critical value of 7.82 in this investigation. Now, because that critical value is as a result of that 7.82, this 7.82, this one is a result of the degree of freedom, right, which is n minus 1. N is number of phenotypes, so degree of freedom here is 3. Okay, phenotypes, we have 4 phenotypes. Phenotype 4 minus 1, which is 3. So our degree of freedom is 3. So it's a product of our degree of freedom and probability, right? The critical probability of 0.05. So that's why we chose it, right? So it is a product of this degree of freedom 3 and critical probability of 0.05. So if you trace it, you can see it. So degree of freedom here is 3. Move it to under, follow it, come to this. This is 7.82. Okay, so product of degree of freedom 3 and critical probability of 0.05. That's why we chose it. Okay, so n is equal to 1, probability of this 1 less number of categories and probability of 0.5. Okay, use your calculated value of chi-square to explain whether the null hypothesis should be accepted or rejected. Okay, suggest a conclusion the biologists could make about the inheritance of the genes controlling wing shape and eye color in fruit fly. Okay, let's solve the first one, explain whether the null hypothesis should be accepted. Now remember the decision model, okay? The same for t-test, right? And same for Spearman correlation. Now this is if this is stated if and only if chi-square calculated value calculated value is greater than chi-square tabulated. Now chi-square table value, that is the one associated with the critical probability of 0.05, that's tabulated value. If it is greater than it, okay, if this is greater than this, then reject null hypothesis. Reject the null, reject null. Reject null hypothesis. So reject it. Okay, the null hypothesis is something like this. Yeah, reject null hypothesis. Okay, reject it, otherwise accept it. So the calculated form, we got 54.44. remember 54.4, okay, 4, and then for tabulated value we had 7.82. you saw it in that table, right? So you can see that calculated value is actually greater than the chi-square value at the critical probability of 0.05, which is 7.482. So reject. So now hypothesis is rejected. Okay, this means that there is significant difference between the observed result and the expected result. There is significant difference. Okay, so The difference could be the fact that the genes that was suggesting that the genes, wing shape and eye color in fruit fly, they are on the same chromosome, so they have autosomal link. They are autosomally linked. Okay, because that is what shows autosomal linkage. They are on the same chromosome, so they are not um, independently assorted. So if this is a chromosome, for example, okay, so these guys are on the same chromosome, right? The two genes on the same chromosome, okay, yeah. So the other one, E, okay, is here. See, so this gene and the other gene, okay, yeah, they are linked, autosomal linked. They are on the same chromosome, so they are sorted. So that's the result of that, right? That's why we see that. See what is expected to the answer to the first one. Reject no application and state that the calculated value is higher than this, the critical value. If no application, of course, is rejected. So between this and there's a significant difference. Difference is not due to chance. Okay. Now there's 5% probability that the difference is due to chance, but there's 95% that is not due to chance. All right. So explaining further, there's a significant difference. Idea that genes are on the same chromosomes are not on. They are not on different chromosomes. They're actually linked. All right, they show autosomal linkage. They do not assort independently. So that's what we said at the beginning. So they are on the same chromosome. So they do not assort independently. Where one goes is where the other one goes. So a chromosome will have this. Okay, yeah, this is autosomal linkage, right? So you see that we're applying paper 4 concept here, right? Now these chromosomes, these genes are linked. Do you get? Yeah. So you see that their recessive form also will be linked. No wonder the result, okay, represents more of the parental class. So this is going to be small n, okay, and because these are sister chromatids identical, this also will be this allele lowercase, and this will be that allele lowercase. Okay. All right. So that's it, guys. 3 marks. So thank you for watching these videos. Make sure you share these videos. Stay with us and get ready for our last series on this, which is series 8. So remember, this is schedule. Bye for now.